Hello, my name is Emily Donnelly, and I'm an undergraduate researcher in Dr. Justin Leglitter's lab. My current project focuses on using stabilized Huntingtin oligomers to determine their mechanism of lipid membrane binding. Huntington's disease is one of many neurodegenerative diseases, in the same class as Alzheimer's disease and Parkinson's disease. These diseases are protein misfolding disorders, which are caused by rearrangements of a protein's conformation, or the way it is folded, to form aggregates, or clumps of protein. The protein that is misfolded in Huntington's disease is Huntingtin. The first 17 of the amino acids that make up this protein are referred to as NT17. A polyglutamine expansion, or a repeated sequence of the amino acid glutamine in NT17, allows Huntingtin to aggregate or clump into several different forms, which can damage membranes made up of lipids within cells. NT17 affects both aggregate formation and lipid membrane binding, so a possible method of treating Huntington's disease could come from blocking Huntingtin's ability to interact with membranes. Previous data from our lab has shown that the oligomer state of aggregation has the highest affinity for membranes. Oligomers are made up of a few protein subunits, while fibrils are long, rod-like structures made up of several subunits. When the lysine residues in oligomers are cross-linked with the molecule difluorodinitrobenzene, or DFDNB, they are stabilized. They do not further aggregate into fibrils and do not interact with membranes. In our current project, we attempted to prove that this stabilization was caused by a change in the flexibility of the oligomer's conformation, not a change in the charge of the linked lysine residues. First, Huntingtin mimic peptides known to stabilize oligomers were mutated so that the lysine residues lost their positive charge and became neutral. The residues affected were K6, K9, and K15. 7 combinations of mutated residues were used: K6, K9, K15, K6 and 9, K6 and 15, K6, 9, and 15, and regular NT17. Oligomers were allowed to form using these peptides and incubated for 8 hours. Samples were taken at 1, 3, 5, and 8-hour time points. The samples were then analyzed using atomic force microscopy, or AFM, to determine what type of aggregates were formed. PDA, or polydiacetylene, and thioflavin assays were also performed. In the bottom left is a diagram of how atomic force microscopy works. A cantilever with a nanoscale tip moves over the surface of the sample, moving up and down over structures on the sample's face. A laser bounces off the cantilever, and its change in position is recorded by a sensor, allowing us to make a sort of topographical map of the sample's surface and determine the shape of any aggregates. The results of the thioflavin assay showed that the cross-linked samples form fewer fibrils than regular Huntingtin. This can be seen in the graph of the assay, where the dark blue line represents unaltered Huntingtin and the other lines show cross-linked proteins. The results of the PDA assay showed that the samples retained their ability to interact with the membrane. A higher colorimetric response in this assay means more interaction with the membrane, and as you can see, all of the samples had at least some interaction with the membrane. The K6, K9, and K6, 9, and 15 samples had slightly higher interaction. Finally, AFM images showed that the samples formed oligomers and did not aggregate further into fibrils. It also showed that they remained stable for the full 8 hours of incubation. The images on the top right show the control, which contains fibrils, and the K6, K9, and K15 samples, which contain oligomers, at the 8-hour time point. This experiment showed that neutralizing the charge of the lysine residues stabilized the aggregates into oligomer form without removing membrane activity. Therefore, we can conclude that the loss of membrane activity in the cross-linked oligomers was due to changes in their flexibility, not charge. While these are preliminary results, we now have more information on the mechanism of oligomer stabilization and can perform further experiments on preventing membrane damage. Thank you for viewing my presentation.